Although it's not proven that nutritional intervention is associated with better cerebral outcomes, numerous observational studies have consistently shown how healthier diets link to better cognitive function. Previous studies in animal models have shown that the accumulation of saturated fats play a role in neurodegeneration. Yet in humans, more research is needed in order to confirm this hypothesis. Here in the Yassine lab, we have found interesting evidence that illustrates a correlation between various fatty acids with total tau and p-tau. You may be asking yourself, what are fatty acids? What is tau and its difference from p-tau? Does the increase of saturated fat in the brain cause neurodegeneration or cell loss? Inside our bodies, fatty acids work as the building blocks of fat, providing as an energy source. They're predominantly made in adipose tissue and the liver. It can influence hormone homeostasis along with affecting the cell and tissue's metabolism and function. Fatty acids are essential in heart health, cognitive function, and in the prevention of cancer and obesity. Tau is a protein that functions to help stabilize neurons by promoting the assembly of tubulin into microtubules. These microtubules are responsible for transporting essential substances to neurons in the brain. P-tau is different from tau as it is abnormally phosphorylated, indicating a more specific biomarker for Alzheimer's disease. When a protein is phosphorylated, it alters its structural conformation, meaning its function can be modified by becoming activated or deactivated. This experiment was designed using two cohorts of older, non-demented individuals. Researchers analyzed the participants' lipid and tau levels in the cerebrospinal fluid. We associated these readings with the participants' total tau and p-tau, finding interesting data. Only 4 lipid species were found in the confirmation cohort, but a significant 37 lipid species in the discovery group. By analyzing CSF in both test groups, we were able to conclude a positive association between saturated phospholipids and p-tau. This data suggests that saturated PC is associated with Alzheimer's dementia pathology via tau phosphorylation, independent of amyloid biomarker status. With that being said, there seems to be a correlation between high levels of saturated fat and Alzheimer's disease. This begs the question of how can we change this accumulation of saturated fat? Is this diet driven? Is the buildup because we are unable to clear them? Or do some people have genetic predispositions that make excess saturated fat? These are great questions that need additional study. This illustration shows the various pathways that lead to the hyperphosphorylation of tau. It is important to further understand these processes because they give insight on what drives neurodegeneration. To find the publication and learn more, please visit yassinelab.com.